Gregor Mendel is an Austrian monk who started this garden pea experiments, and Mendel didn't have any formal knowledge or education about this, about genetics, health, medicine. However, he noticed that different varieties of peas, when they are crossed, they will present differently. But this is the example of hybridization, creating the plant hybrids, and it's not something new that Mendel started, because it's been done during many centuries. But Mendel is the father of this discovery because he was the first one who noticed some kind of law, some kind of regularity in the crossbreeding the plants. So before Mendel, the breeding of the plants and animals was considered rather random process. But with Mendel, who is considered the founder or father of genetics, it's the notions start appearing that there is a rule and that there are Mendelian laws, which you all know, and I won't be spending time repeating here. So, terminology: locus on a chromosome. So locus is the location occupied by a gene on a chromosome, and allele is the alternative version of a gene at the locus. So to understand this, we need to realize that each individual will have two alleles for each gene, and homozygous person, individual, has two identical alleles of the given gene. Heterozygous individual will possess two different alleles of the given gene. So basically, allele, it's the same gene who's responsible for the same trait, but it's different, it's alternative version. And blue eyes, brown eyes, blonde hair, brown hair. So this is controlled by the same gene, but by a different allele. When we coming to the notion of polymorphism, is we are talking about locus that has two or more alleles that occur with frequency that can be appreciated. So we can fairly commonly predict that these alleles will occur with certain frequency. So homozygous genes, so loci on a pair of a chromosome have identical alleles. Okay, so we have all blood type, and both alleles are identical. Heterozygous is loci on a pair of chromosomes have different alleles, and if we're talking of the blood, it's A and B blood type, A and B alleles on a pair of loci. Differences between genotype and phenotype. So genotype, what they have, the genetic makeup of the organism. Genotype is the blueprint. Phenotype is what the individual will demonstrate, will show. So phenotype is what we can see or detect somehow by some kind of measuring device. It's the appearance of the organism or individual. So genotype, basically the example given here, that the person with type A blood can have AA or AO. A can be considered the phenotype, but AA and AO is the genotype. when two alleles are found together and one of them will be dominant, and the dominant allele will be the one that's the presentation of the feature or is observable, the hidden one will be recessive. So if you have two alleles, one is brown eyes, responsible for brown color of the eyes, the other for blue eyes, and brown is dominant in this case and blue is recessive. So when we're talking genetics, the dominant allele is always represented by capital letter and recessive is always lowercase letter. And alleles can appear together and be codominant. And that can be a case of codominant alleles. When the individual has a gene that is not expressed and which is recessive, so that individual can be called carrier. When recessive disease is presented and patient demonstrate the recessive disease, both genes in a pair should be recessive. So in this example, if someone is a carrier of sickle cell anemia, and let's say capital S is no sickle cell trait, while lowercase s is sickle cell trait, so a patient has to have both lowercase s letters to demonstrate the sickle cell disease. Of course, if we have two capital letters S, that means patient neither carrier or doesn't have a disease whatsoever. So a patient cannot or person cannot give the disease to his or her offsprings. Penetrance is another term I would like to talk about it, and penetrance is actually the percentage of people with specific genotype who may express the expected phenotype. So we're coming to the notion of expressivity here, but in this case it all depends how much this patient will have presentation of the disease. So what percentage of the people with the gene with the gene will have the disease? So example, retinoblastoma is incomplete penetrance, which means 10% of affected children with the gene will have no disease. Expressivity is the variation of a phenotype associated with the genotype, and to be more detailed but at the same time clear, we can say expressivity is the rate of presentation of particular disease, disorder, or a trait. This can be caused by modifier genes, and this can vary in degree of how it affects the individual. So modifier genes at other loci can modify the expression of the disease-causing gene. Environmental factors can influence the expression of the disease-causing gene, and also the mutations at the locus. So example is a hemophilia A, one of the examples where we have a mutation that alters just one amino acid at the factor 8 gene, and it will result in milder hemophilia A. If we introduce a stop codon, or stop codon that will terminate prematurely, the translation this may result in more severe form of hemophilia A. So the disease, another disease I want to talk about is the von Recklinghausen disease, which is an autosomal dominant disorder. It affects the long arm of chromosome 17, but the expressivity can vary significantly. It can have just a rather dramatic appearance of the disease. So single gene disorders are autosomal dominant disorder and autosomal recessive disorder. And again, this is just definition. Abnormal allele is dominant, normal allele is recessive. So we just need one allele to express the disease. So it only takes one allele to express this disease, and in this case, if we are talking about autosomal dominant disorder. When we are talking about autosomal dominant disorder, it may present equally in males and females because it's not sex related. Approximately half of the children of affected heterozygotes individual will show the condition. Homozygous affected individuals are rare. So to explain this, homozygous means they have two copies of the allele that carry the disease, that's responsible for the disease. So the probability of it fairly low. There will be no generational skipping, and in some cases the skipping generation is presented as old wife tales, but it truly happens, and it happens in recessive traits. But in dominant disorders, the generation skipping will not occur. To help illustrate this, we need to return to the diagram and our Punnett square, and at this we see that if we have homozygous affected individual, what I was saying before, this is usually rare, means both parents have to have the trait, have to have the gene for this, and they should, then the probability of it is still 25% to have someone with this happen. But having an offspring with the disease, the probability is, as you can see, 3/4 or 75%. So large D represents the disease, the disorder. So only 25% if the offspring will not have the disease. So, but again, it should take both parents having the disorders, having the autosomal dominant trait. More commonly, you will see situation that affected parent means someone with no disease, and at this time we have probability of offspring having the disease only 50%, because one parent will always donate the affected gene, and the other will always donate unaffected gene. So if the affected parent is heterozygous and presents with disease, the affected parents still have one gene that's normal. So that one gene can pair with another normal gene, and let's say disease will be eradicated. However, if affected parent donates a diseased gene, at that time the diseased gene will connect with the normal gene from unaffected parent, and still we will have heterozygous offspring. So to make this short, when affected parent means unaffected parent meets unaffected parent, the probability of having affected offspring is 50%. Again, we're talking only in case of heterozygous affected parent. You can try to do yourself, and if you have questions, please contact me. What will happen when homozygous affected parent, and this is the purple square on this, meets a unaffected or normal parent? You know, you can play the scenarios around. If you have any problem with it, let me know, but this is a good exercise to see what will happen. This is a pedigree picture. Again, a little bit about pedigree. Square represents male, circle represents female, and of course shading, or red in this case, represents the dominant trait. So at this point we don't care about gender because we are talking about autosomal trait. So and this is the pedigree, goes through 4 generations, and you can see that at some point it will be some of the offsprings will not be having disease at all. So the trait will not be there anymore, and you know, it will be lost. But other unfortunate individuals will have the disease. And when we're talking about the trait, it's not only like unfortunate individuals, the disease, but it can be anything, like just features and physical characteristics. So it's not always the disease when we're talking about the traits. It's just trait that's regulated by dominant genes, you know. And autosomal recessive disorders will be again expressed equally in males and females because it's not sex linked. So usually, or most often, the offsprings have parents who are carriers, but they don't have the disease. They are unaware most of the time of the disease, and quarter of the offsprings will be affected, and half of them will be carriers, and quarter of them will be unaffected. In the next slide we will look at it more in detail. For the condition to be expressed, the individual or the patient should be homozygous, so should have 2 diseased genes. So generational skip can be present because you need a donation from both parents. So in the next generation there will maybe just 1. So the autosomal recessive disorders probability can be increased significantly if 2 related individuals mate and have offsprings. It dramatically will increase the recurrence of recessive disorders. The problem with it is that both of the related individuals will have the recessive gene, and if both of them have the recessive gene, the probability of having an offspring with this, with the autosomal recessive disorders, increases significantly, because both of the parents have the recessive gene. So that's why consanguinity presents such a risk for development of autosomal recessive disorders. So when we're talking about autosomal recessive disorders, they are like autosomal dominant diseases, and they fairly rare in the population because they need two recessive genes to connect to create the recessive disorder expression. So here we can see two unaffected individuals meeting, and by unaffected I mean they both are carriers. So in this case, if two carriers meet, still the probability of having the disease in the offspring is much lower than in autosomal dominant disorder, because both parents have to donate the diseased gene to the offspring to have it. So it gives you probability of 25%. Okay. So here in this in the pedigree, eventually these parents will have recessive disorders expressed in their offspring, and it's fourth generation. What's so important in here